Hello students, myself RK Mishra from RK Public School, Gola Bazaar. Here, in this video lecture, we are going to talk about the remain portion of previous video lecture. In previous video lecture, you have learned the definition and discovery of DNA fingerprinting, and little more topic regarding the concepts of DNA fingerprinting is almost clear. Let I revise the previous video lecture and continue to the remain portion of that concepts. In this video lecture, we are going to talk about the DNA fingerprinting and their concept part 2. Actually, in part 2, we will discuss the remain portion of previous videos. So, let us talk about the concepts what we have learned in previous video lecture. Actually, in previous video lecture, you have learned the human DNA has 3 multiplied 10 raised 9 base pair in their genome. It means entire length of single sets of the DNA, there is 3 multiplied 10 raised 9 base pair. From that base pair, 99.9% DNA, 99.9% nitrogenous bases, and their sequence are similar. This is similar. So we can say that 99.9% DNA are same in all people, and only 0.1% of that DNA are unique. Only 0.1% DNA are unique. That 0.1% unique DNA gives the clue for DNA fingerprinting. Listen, the 99.9% DNA are same. That do not give the any kinds of clue because it is similar in all. What are the unique? The 0.1%. As you all know, 99.9% of 3 multiplied 10 raised 9 base pair is very much, while the 0.1% is very less. But that less 0.1% is not too less. It is about 30 lakh. This unique DNA is about 30 lakh. Keep in mind that 30 lakh unique DNA can use for the identification of a person from thousands, millions and from billions people. That actually gives the idea for DNA fingerprinting. So in this video lecture, we will talk about that 0.1% unique DNA. Listen carefully. Actually, as I told in previous video lecture, when you obtain the graph from the 99.9% and 0.1% of the DNA, then there will be 2 peaks obtained, the major peak and minor peak. This major peak represents the similar DNA, that is 99.9% DNA, and this minor peak represents the unique DNA, that is 0.1%. What actually consists this 0.1% DNA? Listen carefully. Actually, this 0.1% DNA consists the heterochromatin repetitive DNA. Heterochromatin repetitive DNA. So let us talk about first what is heterochromatin, later than the repetitive DNA. दोनों के concept को अलग-अलग समझा दे रहे हैं. Listen, what is heterochromatin? First, we will describe about the heterochromatin and later than the repetitive DNA. Actually, when you extract the DNA from any people, the 2 types of DNA may obtain. One is loosely packed and other is densely packed DNA. That is densely packed thread-like structure. This densely packed thread-like structure never code for protein. It is totally the junk DNA. This doesn't code for. It do not code for protein. It never participates in protein synthesis, whether the transcription and translation. While this code for protein, this is loosely packed, and this code for protein. Listen, the loosely packed DNA that code for protein is known as euchromatin. This is known as euchromatin, and the densely packed that never code for protein is the heterochromatin. Most of our DNA are made up by heterochromatin, that is waste DNA or totally the junk DNA. There is no role of that kinds of DNA in our body. in your body, in the body of other human being. I hope your concept regarding euchromatin and heterochromatin is clear. Less than 2% of our functional DNA are belong to the euchromatin. It means 98%, more than 98% DNA are totally wasteful. Among them, there is some DNA that is unique. Not all DNA from heterochromatin are unique. Only some DNA are unique. Let us talk about further. The heterochromatin can categorize as the repetitive and non-repetitive. Similarly, the euchromatin also categorized as repetitive and non-repetitive DNA. So let us talk about what is the meaning of the term repetitive and non-repetitive DNA. Listen carefully. पहले तो इसको हम थोड़ा सा इरेज कर दें ताकि आपको कॉन्सेप्ट समझ में आ जाए रिपीटेटिव का. Listen, suppose that this is the DNA strand. Listen, having nitrogenous base pair sequence adenine, guanine, cytosine, thymine, cytosine. ठीक है. और इसमें क्या है? एडेनिन, गुआनिन, साइटोसिन, थ्यामिन, साइटोसिन. ये स्ट्रैंड 1 है, ये स्ट्रैंड 2. ध्यान देना है. स्ट्रैंड 1 में अगला 1 2 3 4 5 6 अगले 6 नाइट्रोजनस बेस पेयर सीक्वेंस अगर यही हो, एडेनिन, गुआनिन, साइटोसिन, थ्यामिन, साइटोसिन. फिर अगला 6 नाइट्रोजनस बेस का सीक्वेंस यही हो, एडेनिन, गुआनिन, साइटोसिन, थ्यामिन, साइटोसिन. यही बार बार रिपीट हो रहा हो, यही एक 6 ग्रुप नाइट्रोजनस बेस के बार बार रिपीट हो रहे हो. So this kind of DNA strand is known as repetitive DNA strand. So we can say that the repetitive DNA strand can be defined as when the nitrogenous base pair sequence repeat itself many times on the same DNA strand. There will be the repetitive DNA. While what is non-repetitive? This is the 6th group of nitrogenous base as adenine, guanine, cytosine, thymine, cytosine. Next 6 is not similar as like this. The next 6 can you assume as the adenine, thymine, guanine. While next 6 will not be similar with these two. There should be the guanine, cytosine, thymine. It means we can say that none of the any group of nitrogenous base can repeat and match with any other. There is known as non-repetitive DNA sequence. This is the non-repetitive DNA strand. Listen. So I hope your concept regarding repetitive and non-repetitive DNA clear. Listen. The repetitive and non-repetitive may be the part of euchromatin, that is functional DNA, or it may be the part of heterochromatin, that is non-functional DNA. Repetitive DNA, if it will be the part of heterochromatin like this, then it do not code for protein. Actually, no any kinds of heterochromatin, whether they are non-repetitive or repetitive, no any kinds of the heterochromatin can able to protein synthesis. So neither the repetitive nor non-repetitive able to protein synthesis. I hope this concept is clear. While if the repetitive and non-repetitive is the part of euchromatin, then it both able to perform the protein synthesis. So this should be in your mind. Repetitive DNA and non-repetitive DNA, if the part of euchromatin, then they code for protein. That is, if they are part of euchromatin, that they may code for protein. The non-repetitive protein will be very functional protein and very good type of protein. The protein from repetitive DNA are very poor type of protein, but they code for protein. Both code for protein. Now back to the next part, that is the heterochromatin. Heterochromatin consists repetitive and non-repetitive. They do not code for protein. The repetitive heterochromatin DNA called satellite DNA, and this satellite DNA is actually the 0.1% unique DNA. Listen, This satellite DNA is actually the 0.1% unique DNA. So the 0.1% unique DNA that are present in your body, that are present in my body, and the unique DNA found in every kinds of people are actually the satellite DNA. This heterochromatin repetitive DNA is known as satellite DNA. So what condition would you apply for the satellite DNA? The first condition, they should be heterochromatin. They must be heterochromatin. And the second condition, if they are heterochromatin, then they may be repetitive type. अब आते हैं, this is the heterochromatin. This is also the heterochromatin. But only this is satellite DNA, not this is, because this is heterochromatin but non-repetitive. पहला कंडीशन सैटेलाइट डीएनए का क्या होना चाहिए? हेटेरोक्रोमेटिन, और दूसरा कंडीशन क्या होना चाहिए? वो रिपीटेटिव. ये रिपीटेटिव नहीं है, सैटेलाइट है, ठीक है, रिपीटेटिव नहीं है और लेकिन हेटेरोक्रोमेटिन है. ये क्या है? रिपीटेटिव भी हेटेरोक्रोमेटिन है, इसीलिए इसको सैटेलाइट डीएनए बोलते हैं. इट फॉर 0.1% ऑफ द बॉडी पार्ट्स. नॉन रिपीटेटिव है तो सैटेलाइट होगा ही नहीं, रिपीटेटिव है लेकिन यूक्रोमेटिन है, इसलिए ये सैटेलाइट नहीं होगा. ठीक है, एग्जाम में आता है व्हाट आर द डिफरेंस बिटवीन सैटेलाइट डीएनए एंड रिपीटेटिव डीएनए. तो लिखेंगे कॉपी में, रिपीटेटिव डीएनए मे और मे नॉट कोड फॉर प्रोटीन. इफ दे आर पार्ट ऑफ यूक्रोमेटिन दैट मे कोड फॉर प्रोटीन, अदरवाइज नेवर कोड फॉर प्रोटीन. इफ द सैटेलाइट डीएनए, if the repetitive DNA is the part of heterochromatin, that they do not code for protein, while satellite DNA never code for protein because it is the part of heterochromatin. ठीक है, पूरी बातें समझ में आई, पूरी चीजें क्लियर हुई. अब आगे नेक्स्ट हम बात करते हैं सिर्फ और सिर्फ सैटेलाइट डीएनए की. ठीक है, चलो आते हैं नेक्स्ट. सैटेलाइट डीएनए. Satellite DNA that forms 0.1% of the total body parts, total DNA. Listen, satellite DNA can be defined as the long stretch repeated many times that belong to heterochromatin. The class of heterochromatin that belong to satellite DNA having long repeated sequence, that is called as satellite DNA. This satellite DNA actually categorized on the basis of this, categorized on the basis of there are so many categories that you can group together or you can separate the satellite DNA in different ways. Listen carefully. अब satellite DNA को कैसे हम अलग अलग पार्ट में, कैसे अलग अलग ग्रुप में डिवाइड करेंगे. Listen, satellite DNA can be classified on the basis of their length of segments. Listen, satellite DNA can be categorized and classified on the basis of their length of segment. The second important point, the adenine thymine ratio. The third, guanine cytosine ratio. What is the length of segment? The satellite DNA may have the shorter length, may have the longer length. So on the basis of length, there is one group. On the basis of number of adenine, thymine, guanine, cytosine, there are different other groups. The fourth category that we can classify the satellite DNA is number of repeats. Number of repeats. On the basis of number of repeats, what is number of repeats? Listen. Suppose that this is the part of satellite DNA having cytosine, guanine, cytosine. One cytosine, guanine, cytosine. The other will say the cytosine, guanine, cytosine. तो यहाँ दो ही बार रिपीट किया, हो सकता है कि ये चीज 4 बार आई हो. ठीक है, एक बार ये रिपीटिशन है, सेकंड नंबर ऑफ रिपीटिशन और थर्ड हो, थर्ड वाला भी cytosine, guanine, cytosine, फोर्थ वाला भी cytosine, guanine, cytosine. तो ये कई बार अगर रिपीट कर रहा हो तो काउंट करते हैं कि ये नाइट्रोजनस बेस पेयर्स के ग्रुप कितने बार अपने आप को रिपीट कर रहा है. जितने बार रिपीट कर रहे हैं उसी को बोलते हैं हम लोग कॉपी नंबर. So the copy number can be defined as number of repeats of nitrogenous base pair sequence. ठीक है, so on the basis of number of repeats we can say that some satellite DNA has very least amount of repetitive DNA sequence, some satellite DNA has very much amount of repetitive DNA sequence. Clear है इतनी बातें सबको? अब आगे आते हैं, ऑन द बेसिस ऑफ नंबर ऑफ रिपीट्स, ऑन द बेसिस ऑफ नंबर ऑफ रिपीट्स ऑफ नाइट्रोजनस बेस पेयर सीक्वेंस the satellite DNA can be classified further into the microsatellites and mini satellites. Now back to the types of satellite DNA on the basis of number of repeats. Satellite DNA on the basis of number of repeats, number of repeats. Satellite DNA on the basis of number of repeats may be categorized and classified as the microsatellites and the mini satellites. The microsatellites and the mini satellites. There are two class of satellite DNA. The microsatellites and the mini satellites. Listen, what is microsatellites and what is mini satellites? ये याद रखना, ठीक है? Microsatellites can be classified as the part of satellite DNA or the type of satellite DNA having very short stretch repeats many times. That is, when short stretch repeats are found. When short stretch repeats are found, that satellite DNA are called as microsatellites. And when very long stretch repeats are found, that satellite DNA is known as the mini satellites. So microsatellites, when short stretch repeats are found, short stretch and long stretch repeats से क्या मतलब है? Suppose that ये microsatellites है, ये mini satellites है. So microsatellites में बहुत कम नाइट्रोजनस के बेस पेयर सीक्वेंस ये रिपीट्स करते हैं. सपोज दैट ये AGC है, AGC है, AGC है, तो इसमें कितने नाइट्रोजनस के ग्रुप है? 3 के नाइट्रोजनस बेस के सीक्वेंस ये कई बार रिपीट्स करते हैं. ठीक है, दिस इज द प्रॉपर्टी ऑफ माइक्रोसैटेलाइट्स. माइक्रोसैटेलाइट्स. Now back to the mini satellites. In mini satellites, long stretch of repeats many times. Adenine, thymine, guanine, cytosine, thymine, guanine. एक ग्रुप ये बन गया. Adenine, thymine, guanine, cytosine, thymine, guanine. दूसरा adenine, thymine, guanine, cytosine, thymine, guanine. तो क्लियर है कि अगर लॉन्ग स्ट्रेच रिपीट्स करेगा तो बहुत कम बार रिपीट्स करेगा, लेकिन शॉर्ट स्ट्रेच रिपीट करेगा तो ज्यादा बार रिपीट्स करेगा. सो इसमें हम बोलते हैं लॉन्ग स्ट्रेच रिपीट्स मैनी टाइम्स. That is called as mini satellites. लेकिन रिपीट्स होता है, चाहे वो लॉन्ग स्ट्रेच हो चाहे शॉर्ट स्ट्रेच, रिपीट्स सैटेलाइट डीएनए में होते हैं. रिपीट्स ये भी करेंगे, रिपीट्स ये भी करेंगे, लेकिन ये थोड़े से कम कॉपी नंबर में होंगे, इनके रिपीट्स काफी कम मिलेंगे. ठीक है, सो शॉर्ट स्ट्रेच रिपीट्स, लॉन्ग स्ट्रेच रिपीट्स. अब आते कितने स्ट्रेच रिपीट्स करते हैं. इन केस ऑफ माइक्रो सैटेलाइट्स 2 to 6 बेस पेयर सीक्वेंस. अभी कितना बेस पेयर है? 3, यानी कि 2 और 6 के रेंज में मिनिमम 2 मैक्सिमम 6 बेस पेयर सीक्वेंस कितने बार रिपीट्स करते हैं? 5 to 200 टाइम्स रिपीटेड. इन केस ऑफ माइक्रो सैटेलाइट्स 2 to 6 बेस पेयर सीक्वेंस repeats 5 to 200 times. That is the property of micro satellites. In case of mini satellites, the 10 to 60 base pair sequence, 10 to 60 base pair sequence repeats itself 5 to 50 times. It means ये नाइट्रोजनस की बेस पेयर सीक्वेंस 1 2 3 4 5 6 है, ये मिनिमम 10 हो सकते हैं, मैक्सिमम 60 हो सकते हैं, यानी इससे भी ज्यादा बेस पेयर सीक्वेंस होते हैं जो 5 to 50 टाइम्स रिपीट कर सकते हैं. 5 बार हो सकता है या मैक्सिमम 50 टाइम्स रिपीट कर सकते हैं. That mini satellites actually gives the clue for DNA fingerprinting. So basically, which part of 0.1%, which part of satellite DNA are used in the DNA fingerprinting? The mini satellites. The mini satellites actually used for the DNA fingerprinting. I hope your concept regarding today's topic and today's video lecture is clear. If you have any doubt or problems, you can frequently ask in comment box. I will probably able to solve your problems. In next video lecture, we will learn about the DNA fingerprinting and its concept part 3rd. Thank you.